Bouchard is a pioneer in exercise and genetics. He's led the way in this field for quite a long time, and hopefully you'll gain some of that as you hear his talk today. Dr. Bouchard comes to us courtesy of the Texas ACSM lecture tour, and so he's traveling around the state, and he saved one of his best stops for last. Just to let you know about Dr. Bouchard just a little bit, he started out as a physical education major, got his undergraduate degree at Laval University in Quebec, Canada, got his master's degree in exercise physiology at the University of Oregon, and then traveled to the west here, the University of Texas, to get his PhD in population genetics. After his PhD, he returned to Canada and spent the majority of his academic career at Laval University. In 1999, he decided to travel down to Louisiana, become director of the Pennington Biomedical Research Center, where he's located today. In 2010, he stepped down from that position as director and resumed his role back in the lab. He wanted to get back in the lab and get away from administration, focus a little more on his research. And so currently he is a professor and Barton Chair of Nutrition and Genetics. So Dr. Bouchard has a long list of honors and accomplishments, and it's really quite an impressive CV. But I think the thing that the students and the faculty should take note of, the thing I find probably most impressive, if you look at his CV, he has over 1,000 peer-reviewed publications, over 38,000 citations, which means people are actually reading and citing his literature. And his H-index, for those of you who know what that is 91. So it's incredibly high. And so he is not only a productive scholar, but the work he's doing is important and interesting to the field and to others. He's been well-funded throughout his career and is hopefully going to tell us a little bit about some of the work he's doing now and some work that will get funded in the future. So without further ado, Dr. Claude Bouchard.
Thank you, Michael. Well, thanks for the nice introduction. I did all that, but I am 150 years old. And I began at 5 years. Okay. So today I'm going to try to get you excited about the possibility that physical activity may diminish the risk that people may have to gain weight and become overweight or obese. And obesity is a huge problem, as you know. The World Health Organization, to give you just one piece of statistics, estimates that we now have 1.6 billion people who are overweight or obese around the world, 400 million of which are frankly obese. So, and most of them actually are in developing countries like China and India. So these numbers will go up dramatically as they are in their industrial and nutritional transition. I don't know if you can see that well. So this is the David sculpture from Michelangelo. And he spent some time in the US and was exposed to an obesogenic environment. And that was what happened to him. And now we have returned him to his museum spot in Florence in Italy. He's ready for to undergo therapy. But that's a good example of gene-environment interaction. It's a cartoon, obviously, but it's an example of gene-environment interaction because David was lean in his original museal spot in Florence, and then he moved to an obesogenic environment, and obviously he had a genetic predisposition because he began to gain weight, and look at him today. Michelangelo would not be proud of him. I'm going to talk about these topics. First, I will use a few slides to show you the role of single gene defects in the obesity epidemic. And I will use this as to demonstrate that genetics and single genes can have a big impact on the risk of becoming obese. Second, I will move to the common form of obesity, the typical form of obesity that we see on the street all the time, and give you my version of the constellation of contributing factors. Then give you an indication of the magnitude of the genetic component, and I will switch to the current status of our understanding of the genomic predictors, and I will spend most of my time on the point number 5, where we will look at the interactions between genes and activity, and I have some concluding remarks. Okay, the single gene defects. So here we have, I think I'll put it at the right angle so that I can see. Here we have 3 types of single gene defects. Let's begin by those labeled at the top, non-syndromic obesity. So they are on your right here. We have 8 of those, and these genes are causing obesity. Oh my gosh, Joanne, how are you doing? I'm not seeing you in a long time.
I
know. Non-syndromic obesity. Where is the other half?
Oh. Oh. Good to see you.
Good. So these types of diseases are caused by one gene, and obesity is the main manifestation. It's different from the other types that I will describe. For example, let's take leptin. If you have a deficiency in the leptin gene, you don't produce a competent leptin hormone, you become obese. And now we know that's really the case, because when we give leptin to the people who have this deficiency, they normalize their body weight, and they enjoy normal body weight regulation, and they get cured of all their disorders, which are many. So we have learned a lot about the biology of the regulation of energy balance and of adipose tissue biology through these diseases, because it helps us identify pathways and mechanisms. Some of them are more frequent than others. The melanocortin-4 receptor is one which has a lot of DNA sequence variation. Most of them are benign, but there are quite a few of them that diminish the activity of the receptor. Some of them profoundly, some of them marginally. And you see body weight scaled almost to the level of disruption of the receptor. About 5% of the children who are obese, particularly severe obesity, 5 to 6%, have a deficiency in this gene. So overall, about 6–7% maybe of all the severe obesity cases are explained by those. Then we have the syndromic obesity. Those are also Mendelian disorders. And the difference with the preceding class is that here obesity is only one of the secondary manifestations. They have other disorders. For example, they are mentally retarded, or they have locomotor problems, or they have bone anomalies. And obesity is one of those disorders. So it's not as direct, but we know that the deficient genes play some role in the regulation of energy balance. And we've learned a lot about them. For example, the second one at the top, BBS1 to 12, is one that we had no idea what it was before. Now we have identified, I think now it's up to 16. We have 16 different genes leading to Bardet-Biedl syndrome, and the deficiency is in the ciliary molecules that are the surface of a cell. So this is a biology that very few people were paying attention to, but now, as a result of this disorder, we've learned a lot. And then we have the lipodystrophies. Here, these genes are related to the disorder where people have no adipose tissue, or partial lack of adipose tissue in some body segments. And some people go through life with this reasonably healthy, but they tend to have metabolic problems, because when you don't have adipose tissue, you don't have a place where there's a sink for the excess fatty acid that you consume. And that's a problem. You have to store these excesses, and they go in the muscle, in the liver, in the pancreas, in the cardiac muscle. They go in non-storage organs. And that's a big problem. These guys typically are diabetic. Okay, so that's the current status of this categories of genes. Then an important lesson from these diseases is that you may not be obese, you may not be affected, but if you have, because it takes generally 2 copies of the gene, they are mostly recessive disorders, but if you have 1 copy, you tend to have an intermediary phenotype. I will use leptin again to demonstrate that. In Pakistan, Sadaf Farooqi was able to identify 13 heterozygote, demonstrated heterozygote at the molecular level for the leptin mutation that normally leads to leptin deficiency. And she used 95 controls. And as you can see on this slide, those who are heterozygotes have more fat than those who have no copies of the deficient allele. And those who are heterozygotes have much lower levels of leptin than the controls. So I could give you other examples, but this will show the case that carrying 1 copy may be playing a role, and we don't see it because we don't look for those genes in the population, but it may be more prevalent than we think. Okay, now let's move on to the common forms of obesity. So I'll start with my version of the energy balance equation. Of course, we all know energy intake and energy expenditure are driving this, but it's not all. The net result of a given energy intake and energy expenditure conditions can be understood only when we appreciate what happened to the partitioning of energy. And if you have a phenotype which favors fat accretion over fat oxidation or lean tissue deposition, then you set the stage for a greater risk of gaining fat. If you do that, normally you stimulate adipogenesis. You increase either fat cell size, but you also increase the number of fat cells, which is a healthy response. If you don't increase the number of fat cells for storage, you have a problem. You're on your way to diabetes. If you have the ability to increase fat cells, you may become enormous, but it's a healthy response. It's an adaptation that will protect you against metabolic disorder. Okay, if you are in that loop for a long time, you gain weight, and eventually you become obese. Now, all of this is intra-individual. It's within the person. That's how it happens. It's regulated at that level. But we know that there are powerful external forces impacting on this equation, or system, I should say. Social, environmental determinants, physical environment, behavior, and of course, the rest of the biology outside of the energy balance equation. So all of these are studied. You know, those of you who take a peek at the literature on this, you talk about portion size, advertising to children, etc. You know, lack of TV time, lack of ability to exercise, no walking paths, etc., etc. Lots of them. Actually, I have done the effort of putting them together, and that's what it gives. There's a constellation of factors. So when someone tells you, we need to do X, and we're going to resolve the obesity problem, it's compensated so easily by the others. Look at all the numerous environmental and behavioral factors involved. And then it comes on top of our own biology, which puts you more or less at risk for gaining weight. So we're not going to focus on all of that today. I am focusing on the genes, and it will become evident in a minute. let me reorganize that now to make it simpler, and I will use that model for throughout the talk. So let's start again with our within-person system. We know that social and the built environment are important. When they favor obesity, we call that an obesogenic environment. An obesogenic environment typically favors obesogenic behavior on the part of a large number of people. And then this is filtered by our own personal biology. And we have a given set of pressure on this within-person system to regulate energy balance at various levels. Also, we know that the biology feeds back on behavior, and that's ample research has shown that. So let's start examining the biology box and the arrow leading from biology to energy balance in that system. First question is there evidence for a genetic component to obesity? Now, I could spend a lot of time on this. Between the 1970s and 1990s, there were literally hundreds and hundreds of papers published on that question using monozygotic twins and dizygotic twins reared together and apart, nuclear families, extended pedigrees, adoption data. A lot of studies have been done. And we have not resolved that yet. The data don't agree, and that's for a number of reasons. I will not get into that unless you ask me in the question period. But the important thing is to see, is there is the evidence showing that on average there is a genetic component? And I will use the Lambda coefficient to demonstrate my point. It's less controversial than the irritability estimates and other similar parameters. So this coefficient is essentially a quantification of the familial risk. And it is calculated as the magnitude of the increase in risk among biological relatives. Typically, we use first-degree relatives compared to the prevalence of the condition. So, for example, the prevalence of the condition of obesity in the US when this study was done in 1990 was probably around 20%. Now it's 35%. So it's calculated that way. You take, for example, a brother, one of your brothers who is obese at a given level, say BMI of 30, and you ask how many among the relatives have a BMI of 30 or more. And you compare that prevalence in the SIB-ships, in the nuclear family with the parents and the brothers and sisters, you compare that to the prevalence of the population. So this exercise was done in a number of studies. This was the first paper published on that very issue in humans. And it shows that, for example, take a BMI of 30. The increase in the familial risk is about 3. So 3 times more risk if you have a first-degree relative. If you have a BMI of 40, it's more than 5, and if you are really severely obese, it's 6, 7, 8. It goes up. So there is a risk, and the risk is stronger with the increase in severity of obesity. That's all I need. I don't need to know how much it explains. That's all I need to justify going after the genes, hunting for the genes. There are lots of studies that have been done on irritability, and I've summarized some of those here. Some of them relate to BMI, energy intake, fat intake, resting metabolic rate, thermic effect of food, level of activity, energy partitioning. For all of them, based on twin data and family data, we have significant irritability coefficients, but they are not very high. They are generally in the about 1/3 of the variance on average. Okay. First, genomic predictors. So that will now lead me to genetics, really. So this adventure began in 2001 when we had the sequence of the genome, the first almost complete sequence of the genome in 2001. Because now we had a sequence that we could refer to for comparison purposes, but also along with the sequence came the technologies to sequence and to genotype people for variants. It took about 10 years to have that sequence at a cost of about $3 billion. And today we get all of this, if you have some connection with the industry, for about $1,000. So we went from $3 billion to $1,000 in a decade. That's remarkable. And it's going to go down even more. Most of you probably know what is a SNP, but I'm going to say a word about that to make sure you understand that. It is one of these variants in the genome, but it is a special kind. It is when one base of DNA is different between the two versions that you have, one from your mother, one from your father, or between your neighbor or colleagues or other members of the population. To be called a polymorphism, a single nucleotide polymorphism, it must be present in at least 1% of the population. So we call that common variants. And all the data that we have about the common form of obesity are based on common variants. We have no data on rare variants except in single gene defects. I will come back to that. Now, interestingly, in the genome, the first one was sequenced in 2001. By now, there are about 100,000 people who have been sequenced in the world. So we went from 0 to 100,000 in record time, and it's going up exponentially. So now we know that SNPs are prevalent. There are about 10 million of them in Homo sapiens that we know of, about 10 million. But a given person carries about 3 to 4 million. So what has been studied thus far, the genetics of diabetes, atherosclerosis, cancer, dementia, all of these studies are based on these common SNPs, are based on the 3 to 4 million that we carry in our genome. So there are limitations because we have a lot of other variant features in the genome. I will mention that in a second. So these SNPs for a given person, we can describe them. That's not an exhaustive list, but I have highlighted a few here. For example, we have up to about 11,000, each of us, 11,000 SNPs that translate into a change in amino acid in the proteins that this message encodes. So they are non-synonymous, about 11,000. About 100 maximum premature stop codons, which means an incompetent peptide, if that message is coding for a peptide. We have about 50 splice site disruptions, which means that we will not remove the introns in an orderly manner, therefore we'll have an incompetent messenger RNA. We have up to 250 on average insertion in the DNA of a message. New bases have been added there, or some have been deleted, about 250 each of us. We carry a lot of copy number variants. So there are plenty of sequences in the genome that are repeated, sometimes 3, 4 bases repeated 10 times, 100 times, 1,000 times. And some of us may have only 500 copies of it, others may have 10,000 copies of it. That is not that dramatic most of the time, but sometimes it interferes with the expression of the genome, and there's a growing number of diseases that relate to these copy number variation. Here I have highlighted only the repeat segments where the length of the segment is 450 bases and more. And we have 1,000 sites where we vary in the number of copies that we each carry. So that's more important because it's not a small fragment, it's a big fragment, and it can interfere with expression. More importantly, we have, each of us, about 200,000 to 500,000 rare variants that we have inherited from our parents, and it's not in our remote ancestry, it's recent. These variants are not in high frequency because it's too early, or they may have large effects and they are not very favorable, so the forces of selection have not yet eliminated them from the pool. They're there, you carry them. they potentially have large effects on the phenotype. So we carry a lot of those. These have not been studied. The tools that we have used for exploring diseases do not provide any information on them. And finally, about 2 weeks ago, there were a series of about 30 papers published in the best journals that we have in science, including Science, Nature, Cell, and Genomic Biology, etc. We now know that there are millions of switches regulating the expression of the genes in the so-called junk DNA, the non-coding DNA of the genome, which is 97% of the genome. So I have a slide on that. It's really extraordinary as an advance, and it just happened. So this is the clan model. At the top, we have the blue pedigrees. These are pedigrees, so about, say, hundreds of generations ago. There were mutations introduced, and these mutations disappeared. Some of them were favorably selected. When they were selected, they tended to have small effects, and their frequency has therefore, and they were favorable, and their frequency has therefore increased over time. As we go down, the frequency diminishes, but the effect size tend to go up. And when we go to the remote ancestry, we see that some of these are present only in some family lines and not in others. They may be de novo mutation, actually, and they may have very large effect. Selection has not had the time to eliminate them. They are very difficult to study because it takes large populations, unless they cause a disease. If they are a single gene disease, they will be picked up much more quickly. If they just increase the susceptibility to obesity in, say, 300 people on Earth, it's going to take a long time before we pick them up. Okay, so that we know very little about, and nothing concerning obesity. Now, this is what was published 2 weeks ago. The Encyclopedia of the Human Genome, as it is called. This is the project ENCODE, funded by the Genome Institute at NIH, 32 laboratories in 5 countries, more than 400 scientists, 10 years of research, $250 million. They were exploring that their task was to tell us what is in the dark matter of the genome, in the region that is known not to encode proteins. And that's what they did. And first they discovered that it's not 3% of the genome which is transcribed, but it is 80% of the genome which is transcribed. They confirmed that the number of genes coding for protein is about 21,000. Not 100,000, 60,000, 40,000, 30,000 as we thought. It's 21,000. But we have about 3 million switches distributed across the genome where small RNAs or long RNAs or proteins, transcription factors, bind to impact on the expression of these 21,000 genes. So we take about 25 times more space for the regulation compared to what it takes to code for the proteins. An enormous machinery. And we have lots of small RNAs, lots of long stretches of RNAs, 1,800 transcription factors. They use 8% of the genome as binding site. Promoters, they identify 70,000 promoter regions for 21,000 genes. So each gene has multiple promoters. 400 enhancers regions that augment or diminish depending on whether it's a repressor or an activator that's binding the expression of genes. And this is interesting. If you look at the DNA bases maintained by NIH, you see often that this sequence is a pseudogene. Well, they look at the pseudogene and they say, no, that's not the case. There is no such thing as a pseudogene. These genes exist, but they are active only in some individuals, not in others. We didn't know that in the same cells. only in some, not in others. And they distinguish cell types as well. So an enormous amount of work, and we now have a good blueprint to see what does that mean in terms of function. I could speak about that for an hour. There's so much there. But I just want to get you excited, those of you who study genetics, to get into this. Okay, where do we stand? This is the most comprehensive paper. It was based on 249,000 subjects, and the goal was to identify loci associated, SNPs associated with obesity cross-sectionally with such a power level that you begin to have very small effect size genes. Enough power, statistical power, to pick up the small effect size. So the paper identified a total of 32, 18 of which were new in that particular publication. Oh, now here I have a problem. Every time I click on this slide, it gives me a website. There he goes. I know that's the problem. I need to do that. Yeah. So here we have the results. All the red and blue and green, they are the 32 loci that relate to obesity. Note that this one has a level of significance way, way above the other. The Fto gene. It was the first identified 6-7 years ago, and it's still the one coming out in all the studies. And look at here, this is the p-value. So it was significant at about 10 to the minus 60. So it is significant. Here is 10 to the minus 10. So we're not talking here about 0.05 or 0.01 or 0.001. It's really a lot of solidly established associations. Okay. So what does that mean in practice? So we have 32 SNPs. At each SNP, we have 2 alleles. So the authors have done, this is pretty standard in the study of the genetics of common diseases. You take every SNP and you ask which one of the alleles is associated with, in this case, obesity. And you recode the SNP. If you have 1 allele, you have given a score of 1. If you have 2, a score of 2. If you have 0, 0. So the range that you can have in a global genomic predictor score will range, in this case, from 0 to 64. That's the upper limit of the risk, and those who are less at risk have 0 obesity-promoting alleles. So they found that in their population of 8,000 here, a subpopulation, there were a number who had 21 or less. Was about what number of individuals? So probably about 200 at 21 or less, and about 150 at 38 and more. And when you look at the BMI, you see the red line. The difference between those who had a low genetic score, low genetic predisposition, was about 25.5, and those who had more were around 28 and a fraction. About 3 BMI units between the extremes. So that begins to, separate those who are at risk and from those who are less at risk. It's not the last word, of course. It's not a 15 BMI unit difference yet, because you need a much bigger sample size so that you have more people below 20 and more people above 38 to be able to capture the extreme. It probably will take a couple of million people to do that, so it's not a small study. That's my compendium of all the genes that have been identified thus far. The 32 that I mentioned are there, plus those that relate to waist circumference, waist-to-hip ratio, and the visceral fat measured by CT scan. They're all there. So this is the panel. Only 3 of them, those in red, are also found in the single gene disorder family. Only 3. Melanocortin 4 receptor, PCSK1, which is a convertase enzyme involved in the maturation of the peptides, and a brain-derived neurotropic factor, BDNF. Only 3. All the others are unique to the common garden variety of obesity. So we have a lot more work to do because the predictive power is still low. And we don't know whether it's only a matter of getting the genes with the small effects or whether we have to go to rare variants, copy number variants, microRNAs involved in regulation. I guess we have to do everything because we will have only a partial answer if we tackle only the common variants, however powerful the study is. Okay, so now the central theme, interactions between genes and activity. This is an interesting group. I don't know if you're familiar with it. This is an international federation. It's called the International Federation of Competitive Eating. And the US is a prominent member. And they are trying they have petitioned the Olympic Committee to become an Olympic sport. Many times they have done that. They have not succeeded yet. But what fascinates me is this. Here we have someone, her name is Sonia, who holds many world records. Look at that. 46 crab cakes in 10 minutes. 8 pounds of Vienna sausage in 10 minutes, etc. That's a lot of food. But look at her. She weighs 105 pounds. Amazing. So I guess Sonia has a lot of energy dissipating pathways and is extremely non-efficient metabolically, gets rid of excess calories to be able to remain an attractive woman while performing these incredible feats. So that's an example of interaction. Now let's study that a bit more scientifically. Let me give you my definition of a genotype-environment interaction applied to physical activity. So such an interaction effect is present when the response to physical activity. Here we're talking about regular physical activity exposure, and it could be leisure time, occupational, it doesn't matter, is conditional on your genotype. So the response is determined by your genotype. So you don't see that very well if you study one person. You need to have people with different genotypes. You need to have what we call Mendelian randomization to be able to compare genotypes exposed to the same treatment to tease that out. That's complex and generally very expensive. Most of our studies, including the one I just showed you on the 32 genes, are the result of efforts to quantify what we call the main effect of a gene. Now, the main effect of a gene is illustrated here. I've used adiposity versus activity level since we focus on activity. Let's take the FTO, for example. We have 3 genotypes, 2 alleles, 3 genotypes. This one is protective, intermediate, and this one is at risk for obesity. The main effect, and if there is no interaction, we have 3 parallel lines, and we quantify the average effect. That will be the average of the genotype. It will be somewhere in the middle of this will be the average effect. Plus the environment, which all the lines are going down, so that's the environmental effect. And when we will report this, we'll say the average effect size will be 3%, 5%, 10%, 15%. But we deprive ourselves of a lot of information when we do that. For example, this is a real-life example in which activity was measured by accelerometer for a week. And we have here, in this direction, it's more and more sedentary, and here it's more and more active. The 3 genotypes of FTO, so you see them here, in the inactive state, BMI of about 29, and the other genotype about 26. Big difference. But increasing amount of activity, actually, the genotype at risk become the leanest, or at least comparable to the other two, BMI of 25 versus about 26. So that's a strong interaction effect. Something like this. So if we had quantified not only the main effect, but the interaction effect, we would have a larger effect size, a much bigger contribution to the obesity variation. And, but to do this, it's very difficult to do in a credible way with cross-sectional data, observational data, because you have a lot of confounders there. You need experiments. You need to take the activity level and change it. Then you see appearing the differences between genotypes. If you just ask the question, are you active or not, and you try to analyze that, you're not going to get a clean trait to work with. So that's why there are very few studies that have done that properly, because it has to be experimental and with all that entails. But we have proof of concept that it is true. This is a study, we discussed that briefly this morning with Tim. This is a study that I did many years ago, as you see, published in 1990. So we were doing it actually in the lab 25 years ago. We took identical twins, 12 pairs of them, lean, BMI, mean BMI 22. They came from families where there was, pardon me, no obesity, so that we would not study people at risk. And they had to be available for 120 days, 24 hours a day, 4 months. So they had to be unemployed. We recruited them from Newfoundland to Ontario in Canada. We got them. And the first 2 weeks, we said, okay, you're now captive for 120 days. The first 2 weeks, your only job is to eat what you normally eat. We don't want you to gain an ounce or lose an ounce of body weight. And we calculated the energy cost of weight maintenance and with these data, measuring them every day. And then we said, okay, now you need 2,500, 2,050 calories a day to be weight stable. From beginning tomorrow, you're going to eat 1,000 calories more than that. 6 days a week. On the 7th day, you will eat 2,500. We give you back the energy cost of weight maintenance to help you tolerate this. And we did that for 100 days, and then we remeasured them. So the mean weight gain was between 7 and 8 kilograms. But look at this here. So this is here a brother, and here the other brother. So one dot is a pair. So we have here a pair where the weight gain was 4 and 5 kilograms, and here 7, 8, and then here 12, 13 kilograms. A threefold range in the weight gain. They were controlled 24 hours a day. We studied them 4 pairs at a time, not all 12, because it would have been very hard to control them. 4 pairs at a time. And we paid them well. So that shows that there is something like this. For some people, there is an ability to buffer excess calories in a much better way than some other genotypes. The twin line here is a proxy for genotype. We did not have the gene at that time. We did not have any gene for obesity at that time. It was the proxy for the genotype. We did also the opposite experiment, negative energy balance with exercise, etc. I'm going to show you one that comes from another lab, which helps always when other labs find the same thing as you do. It came from Prague in the Czech Republic, where they put the obese female twins on a 28-day very low-calorie diet in the metabolic ward. They had 14 pairs of them, and the energy consumption was about 400 calories per day only. They were obese, so that was an intensive weight loss program. And look at the results. Here, same model, twin A and B. A dot is a pair. So these were low loser, average, and these were high pairs of losers. very big family line. So clearly, that's a proof of concept to me. It exists. There is a genotype, lifestyle, genotype, calories, interaction effect. Okay, now let's look at the evidence for the interaction with physical activity. The first few studies that have been done have focused on FTO, because FTO is the big gene, so everybody wanted to see whether you could influence the effects of FTO and modulate it with a low or high amount of physical activity. So that's the largest one published recently. I think we were about 150 authors on that paper, so many cohorts involved. And interestingly, Ruth Loss is one of the leaders in that field now, and Ruth was one of the students who started with me at Pennington. She did a postdoc for about 3.5 years with us at Pennington. She's very, oops, another one that will play a trick on me. I need to remember which slide to do that. So here we have the FTO variant, this variant, and here we have the risk of obesity per FTO allele, risk allele. So in the inactive, there's a 30% increase in risk per FTO allele. So we have 2. So the maximum increase in risk will be 60% if you have both. And then in the active, it's 20%. So it'll be about 40% total. And these are pretty good numbers for such a test. While here, it's the increase in BMI. And you have the same thing. In the inactive, you have a higher increase in BMI per FTO allele than you do in the active. So there is an interaction. The response, well, not quite a response because it's not experimental, it's cross-sectional. But the difference is in the expected direction. Activity diminishes the impact of the FTO obesity-promoting alleles. And the reduction is here. On average, in that paper, we came up with a conclusion that about 27% of the risk is the effect of physical activity. That's pretty good. It's true in African Americans and in whites. How much time do I have?
About 5 minutes.
So, believe me, it's true in African Americans and in whites. It is true when physical activity is directly measured instead of only obtained by questionnaire. I showed you that slide before. Very strong. Actually, the effect is stronger, apparently stronger, when it is measured. So there's more research needed on this, but it's certainly suggestive. And then, instead of using only FTO, in this paper, the interaction effect was studied in terms of 12 genes, 12 SNPs instead of 1. So we had a better definition. Oh, again. I wonder why. So we had 12. So therefore, the low genetic predisposition will be, the lowest will be 0, and the highest will be 24. In practice, we have subjects who had 6 or less, and enough subjects who had 17 or more to do these analyses. Here you have the regression line for those who were active. So the whole BMI on genetic predisposition score, and here it's the inactive, and you see clearly that with increasing predisposition, that they separate, and there's a greater gain of BMI with increasing predisposition. But activity diminishes that substantially. In that particular study, they also were able to show over time, over about 3.5 years, what is the change in BMI by predisposition score. So here we have the change in BMI per allele. In the inactive, you have a gain of BMI. Now that's a small value, 0.003 of a BMI unit, but it's per allele, and we have 24 potentially here. In the moderately inactive, it's neutral. In the moderately active, it tends to be negative, and the active, it is frankly negative. So over 3 years, they experience a decrease of BMI per risk allele. So activity makes a big difference. And in this particular study, the diminution, the decrease in risk was about 40% compared to the inactive people. Now, we have no study as of yet on the larger panel of genetic risk of 32 SNPs. So if I go back to my model, then we need to modify this. This arrow is not unique. There are several versions of that arrow. We're not buffering the environment the same way. So we have people who have a big buffer, those who have a moderate buffer, and those who have a very small buffer. And that modulates the risk of being in positive energy balance over time. Okay, concluding remarks. I wish I had time to speak about that, but suffice it to say that we have very few studies where these genes are examined, where the level of evidence is strong, like experimental interventions and randomized controlled trials. These will come, but thus far we're more on softer evidence like cross-sectional studies and longitudinal observational studies. This evidence relates not only to leisure time physical activity or sports participation. It also relates to sedentary time, to occupational activity. I think it's activity at large. It's not strenuous physical activity. And there's, another one. Plenty of reason why we should use the message that potentially all kinds of activity can diminish the risk of a high predisposition to obesity. Here we explored with Tim Church and colleagues the change in the prevalence of sedentary, light, and moderate intensity occupation in the US over a period of 50 years to a number of data banks. And as you can see, moderate intensity activity for occupational activity is 3 METs and more. It has gone down from almost 50% to about 20%. Huge decrease. And what has gone up is the sedentary activity and the light activity. When we convert that into energy expenditure, we have a decrease of about 100 to 150 calories in men and in women as well over that period of time. Well, that can be sufficient to interact with that predisposition and modulate energy balance. It's not only sport, as I said earlier. In addition, I have not said anything about epigenetic mechanism and in utero environment and matching your in utero prenatal environment with the environment to which you are exposed later in postnatal life. But these are important. We need to recognize them. They are here, and they also impact on this. Not only the genes and the sequence differences, but the way the genome is impacted and the way your biology and hormone sensitivity is impacted during in utero. For the sake of comprehensiveness, I wanted to show you this. Finally, last slide, Tim, last slide. This exercise, the purpose of these exercises is to put us in a position to predict, to identify, recognize who are those who are more vulnerable, those who are least vulnerable, and then to see whether we can tailor prevention, public health, treatment to their existing predisposition. So ideally, we would like to take advantage of all the omic technologies. We are currently moving ahead full speed with the genomics, and we have the tools, and we know that it will be done. We'll be able to identify the genomic predictors. It may not be sufficient, but we will be able to identify all those elements that are predictive of, in this case, the risk of obesity. To those, we would like to be able to add the epigenomics. That's going to be complicated because epigenomics, especially in people, in animal, not a problem. In people, you cannot do that and be scientifically strong if you do it from the blood. You want to do it from the tissues that are likely critical here. And what are the tissues critical for obesity? Well, first of all, they are up here in the hypothalamus and other central organs regulating energy balance. That ain't going to happen anytime soon, not even in Texas. And the liver, muscle, adipose tissue, that's more possible, but liver and brain, it's not going to happen easily. Transcriptomics is also potentially very helpful in predicting. But again, it has to be done in the right tissue. It can be done in muscle, in adipose tissue, but not in the other tissues. Proteomics might be helpful. We have the tissue limitation again. Metabolomics, though, looking at the end product of catabolism in the blood and urine, yes, that's possible. So at least combining genomics and metabolomics, we have a good chance of developing a set of predictors for practical applications. Epigenomics, transcriptomics, and proteomics will probably stay in the research domain for a long time. But genomics and metabolomics may move into personalized medicine, into an environment in which we use information to advise people as to what is best for them, how to best address their individual vulnerabilities, avoid behaviors that may put them at risk for disease, and find alternative strategies when they develop adverse responses. On this, I will conclude, and thank you very much for your attention.